Early in this chapter, we talked about the idea that bacteria reproduce by asexual reproduction. Essentially, mitosis is a bit of asexual reproduction. It is cells reproducing a genetically identical daughter cell within a living organism. So this asexual reproduction, as it applies to mitosis, is not producing new organisms. It's producing new cells within a living organism. But the process of meiosis, which is our other type of cell division, is a type of cell division that prepares an organism for sexual reproduction. In sexual reproduction, two gametes, remember that gametes are our sex cells, our sperm and our egg, combine to produce a fertilized cell called a zygote. This zygote is what will divide by mitosis to produce an organism. So if we're talking about humans, a sperm fertilizes an egg. That fertilized cell is called a zygote. That zygote is going to divide and divide and divide until we get, or it is an embryo that is going to develop into a fetus and then into a baby. So meiosis has the important job of producing those reproductive cells, those gametes, for the process of sexual reproduction. So the goal of meiosis is to produce those sperm and eggs. Now, one of the important goals of meiosis is to produce what we call haploid gametes. Now remember, we talked about diploid a little bit ago, where ploid means number of chromosomes. A haploid gamete is denoted by the letter N. Remember, N is your number of chromosomes. The word haploid comes from the word hapless, which in Greek means single. So when we say that a cell is haploid, we are saying that they have one copy of each type. So if we are a female producing an egg, we are going to have one copy of each type. And if we are a male producing sperm, we're going to have one copy of each type. So if we are a human, and you and I are all humans, our haploid number for our sperm and eggs are each going to be 23, where N is the number of chromosomes. We have one copy of each chromosome. So to think about this in contrast to diploid, we could think about this as 1N equals 23, one copy of each type of chromosome. Then when fertilization happens and we have N equals 23 egg and N equals 23 sperm combined together, we now have two copies, or a diploid fertilized egg, two copies of each type of chromosome for a total of 46, where half of our chromosomes came from mom and half of our chromosomes came from dad. So in order to produce a new organism, we start out with the process of meiosis, and then fertilization restores our diploid situation. And it is those diploid cells that divide by mitosis to produce an organism, in this case, a cute little baby. So in fertilization, those two haploid cells, where n equals 23 plus n equals 23, come together to produce our diploid individual, where each cell has two copies of chromosomes for a total of 46. The distinction between whether or not a cell is haploid or diploid is really important. Our gametes are the only cells in humans that are haploid. Our somatic cells, all the other cells, are diploid. All right, so mitosis has two important endpoints. One is to start with that diploid cell and reduce the genetic material by half. So in meiosis, we are going from diploid to haploid. We are going from two copies of each type of chromosome to a single copy of each type of chromosome. So in meiosis, we are going to reduce our chromosome numbers. The other important outcome of meiosis is that it produces gametes that differ from one another. At the end of meiosis, we are actually going to produce 4 daughter cells. Remember, in mitosis, we produced 2. But in meiosis, we're going to produce 4 daughter cells that are all genetically different. They are different from the other daughter cells, and they are different from either of the 2 parent cells. Those genetically different gametes are the reason for variation among brothers and sisters in families. They're also the reason that parents and children resemble one another, that brothers and sisters resemble one another. So they are the way that we pass on genetic information, continuous genetic information from parents to children, but they're also the way that we shuffle the genetic information in a parent and in fertilization in order to get siblings that are just a little bit different from one another. So in meiosis, we are going to reduce our chromosome counts, and in doing that, we actually produce 4 daughter cells, but they are all genetically different from one another. So meiosis takes place in the gonads. The gonads are the cells that give rise to gametes. So our gonads for males are testes, where we produce sperm, and the ovaries, where we produce eggs for females. So our gonads are basically our organs that generate those gametes, those sex cells. Meiosis begins with a diploid cell, a cell where in humans 2n equals 46. And in the end reduces to haploid, or n equals 23. Those diploid cells, those cells that are 2n equal 46, are made up of half of their chromosomes from mom, half of the chromosomes from dad. There are actually 23 chromosomes, or 23 types of chromosomes. One of those types are sex chromosomes. the X and Y chromosome. And we'll talk about those a little bit later. So before we move on and actually talk about the steps in meiosis, I want to remind you of homologous chromosomes and sister chromatids. Now, I talked about this in one of the earlier videos in this chapter. Remember that homologous chromosomes are those chromosomes that contain the same genes in same location. One comes from mom, one comes from dad. So gene A might be here at the top of the chromosome, and mom might have the big A version. Dad might have the little a version. Farther down the chromosome, we might have the B gene, where mom has the capital B and dad has a lowercase b. Maybe down here at the bottom, we have the D gene, where mom has the lowercase d, dad has the uppercase. So we encode the same genes on homologous chromosomes, but the alleles may be different between the two of them. In DNA replication, we produce sister chromatids. So sister chromatids are identical copies of each of the homologs connected at the centromere. They are made by DNA replication, and we saw these in mitosis. They were separated in mitosis so that each daughter cell could get one of the original homologous chromosomes. So keep those in mind as we go through meiosis. So one of the major goals of meiosis is to reduce the genome by half. So let's say that we start out with a cell that is 2n equals 16. So our diploid count for that cell is 2n equals 16. If we reduce our genome by half, at the end of meiosis, we are going to be haploid where n equals 8. Let's say, for another example, that we are an organism where the diploid is 2n equals 36. At the end of meiosis, our haploid number is going to be 18. Let's look at this the other way. If we are an egg, that is n equals 4, and a sperm, that is n equals 4. At the end of fertilization, we are going to produce a diploid cell that is 2n equals 8. Remember, each species is characterized by its diploid number. So humans are 2n equals 46. Anything that has 46 chromosomes is going to be a human. All right, so the goal of meiosis is to reduce our chromosomes by half. I really love this diagram because it outlines very simply what's happening in meiosis. In interphase, we start with our homologous chromosomes, where one came from mom and one came from dad originally. So think about this as in your gonads. You are producing a sperm or an egg, depending on whether or not you are male or female. So you produce a sperm or an egg. In preparation for meiosis, DNA replication occurs, and we produce our sister chromatids. In meiosis 1, we see the first difference between mitosis and meiosis. In meiosis 1, homologous chromosomes separate. This never happens in mitosis. The separation of homologous chromosomes is unique to meiosis. and is what causes the reduction of chromosome number. So if we start out here, 2n equals 2. The beginning of meiosis 1, we are still 2n equals 2 because remember we count our chromosomes based on the number of centromeres, even though replication has occurred. At the end of meiosis 1, when we separate those homologous chromosomes, we are now haploid, where n is equal to 1. So in meiosis 1, that's where we go from diploid to haploid. Then meiosis 2 is actually similar to mitosis because in meiosis 2 we separate sister chromatids, which is what happened in mitosis. So without all of that messy business, we start out in interphase, we copy our DNA to produce our sister chromatids. In meiosis 1, we reduce chromosome number by separating homologous chromosome, by separating homologs. In meiosis 1, we separate sister chromatids. Remember that in meiosis, we are going from diploid to haploid. Also remember that in meiosis, we are producing 4 daughter cells. This is because we have 2 cell divisions. So let's walk through the steps of meiosis. Meiosis consists of 2 separate cell divisions. The first division is called meiosis 1. This is often denoted as M1, meiosis 1. You'll notice that the steps are very, very similar to what happened in mitosis. The general process is the same. We're going to condense chromosomes. Our spindle fibers are going to form attached to our chromosomes, line them up at the metaphase plate, and then pull them apart. But there are a few key differences. Remember that meiosis 1 reduces chromosome number and separates homologous chromosomes. Now, before those homologous chromosomes can separate, homologous chromosomes pair up. So in interphase, our chromosomes begin to condense, they replicate, they get ready for cell division. And in prophase 1, our first phase in meiosis, our homologous chromosomes are going to pair up. They are going to produce something called a tetrad. Tetrad because tetra means 4. We'll talk about what actually happens in crossing over just a little bit later. But all of our homologous chromosomes line up together. Then our spindle fibers, just like in mitosis, will attach to our centromeres and pull those chromosome tetrads to the metaphase plate. Now I want you to notice that we have our sister chromatids here, and they are lined up in homologous pairs. If you need a reminder about what homologous and sister chromatids are, make sure you go back and do that. So you'll see here that we've got all of our tetrads lined up along the center, and then our spindle fibers are going to begin to pull our chromosomes away from the metaphase plate. So in metaphase, our homologous chromosomes line up together. That did not happen in mitosis. All of the chromosomes lined up in a straight line in mitosis. But in metaphase 1 of meiosis, the tetrads move together. And then it is the homologous chromosomes that are separated. So our tetrad is broken up and our sister chromatids move together to the poles of the cell. So the process of meiosis is similar to the process of mitosis, but there are those key differences. We get our tetrads formation, and then we get our chromosomes lined up with their homologous pair at the metaphase plate, and then homologous chromosomes are separated at anaphase 1. The remainder of meiosis 1 is just telophase and cytokinesis. There's no difference from mitosis. So at the end of anaphase, this was anaphase, this is continued from the last slide. At the end of anaphase, our chromosomes decondense. Telophase, in telophase, our nucleus begins to reform, cytokinesis occurs, and we produce 2 daughter cells. So at the end of meiosis 1, we have 2 daughter cells. And we still have sister chromatids. So then, at the end of meiosis 1, we enter meiosis 2. Now, meiosis 2 is basically identical to mitosis. Meiosis 2 is basically identical to mitosis. In meiosis 2, our spindle fibers form, again in prophase. They grow out, attach to our centromeres, pull our chromatids to the metaphase plate. Then they separate our sister chromatids in anaphase. Telophase and cytokinesis occurs, and we produce daughter cells. This is where we get our 4 daughter cells, because each of those cells from the end of meiosis 1 goes through meiosis 2. Remember that the end of meiosis produces 4 daughter cells, all that are genetically different from one another. And this is one of the major goals of meiosis, is that we produce genetically different cells. So in summary, the whole point of meiosis is to prepare for sexual reproduction. It consists of 2 rounds of cell division that reduce chromosome number and produce 4 daughter cells. Meiosis is specific for our production of gamete cells because we want to reduce our chromosome number in preparation for fertilization. So if we're keeping track of differences between mitosis and meiosis, meiosis produces 4 daughter cells, mitosis produces 2. The cells at the end of meiosis are haploid, where at the end of mitosis, they are diploid. And at the end of meiosis, let me finish my thought. At the end of meiosis, we have genetically unique cells. Mitosis, we have genetically identical cells. So be sure, now would be a good time to practice with some chromosome numbers. Be sure that you understand where mitosis, meiosis, and fertilization all change chromosome number. So we said that the end of meiosis produces genetically, I apologize for that, produces genetically unique cells. But we didn't talk about how those cells become genetically unique. There are several sources of genetic variation, which is one of the important aspects of sexual reproduction. Remember that asexual reproduction produces genetically identical cells to the parent cells. So when bacteria divide asexually, the offspring are exactly the same as the parent. But in sexual reproduction, one of the advantages is that you get genetic variation. So one source of genetic variation is mutation. We saw that in early in this chapter when we talked about DNA replication. That genetic variation from mutation does also occur in asexual reproduction. But in sexual reproduction, you have unique sources of variation due to the process of meiosis and fertilization. So one source of genetic variation is sexual reproduction itself. You get different alleles from two different parents. So each parent donates his or own genetic material. That genetic material gets shuffled, and you get unique children that are genetically a combination of each parent. That's why you have characteristics of both your mother and your father. But that's also why your brothers or sisters look similar to you but have different combinations of your parents' characteristics. So let's talk about the other two, crossing over and random assortment, in a little bit more detail. One of the most important sources of variation in meiosis is crossing over. Crossing over is also called genetic recombination. Crossing over occurs in prophase 1, where homologous chromosomes form tetrads. So before homologous chromosomes can separate, before we can reduce the number of chromosomes in our gametes, those homologous chromosomes have to pair up. So remember, we have sister chromatids from DNA replication. They are joined together. They basically pair up by a process called crossing over. In crossing over, your homologous chromosomes come together and they actually connect at their ends in a process called crossing over. In crossing over, they actually exchange part of their double-stranded DNA, and this serves two purposes. One is to hold chromosomes together because we want to keep them together so that they can be separated correctly. The other is to produce that genetic variation. So these chromosomes come together. They actually physically swap DNA, and in the process, they're kind of attached for a while. They move together to the metaphase plate in meiosis 1, where they line up together. When they separate in anaphase 1, the result is that they have swapped some genetic information. So crossing over is essentially a way to produce new combinations from mom's chromosome and dad's chromosome. So we don't get new information. We just shuffle the old information. The result at the end of meiosis is 4 different gametes. So if we were to think about each of these chromatids as their own, going into their own gamete, you can see that they're all different. The one that was on the outside of that tetrad is actually the same as one of the parents. The other one that was on the outside is actually the same as one of the parents. But the 2 in the middle that were part of that crossing over event have now shuffled their chromosomes. So now we have new alleles in the same alleles in different combinations. We have new combinations. So if we're thinking about this crossing over at 23 different chromosomes, you can imagine that there's all kinds of shuffling going on. So crossing over is really important for creating new variation, new combinations of alleles that are unique, that never existed before. It doesn't produce new alleles. New alleles come about by mutation. The only way that we get new alleles is mutation. But crossing over shuffles and produces unique combinations of alleles that were already there. That variation is important for evolution, and we'll talk about why that variation is important once we get to chapter 10. Another important source of genetic variation comes from random assortment. Now, I like to think of this as random lining up at the metaphase plate. So in crossing over, our tetrads line up together. They cross over. They make unique sister chromatids. But at the metaphase plate, random assortment means that the chance of getting this green one with this blue one is the same as the chance of getting this green one with this blue one. So you can line these chromosomes up in any combination of light, dark, light, dark, light, etc., with the light and the dark representing mom and dad. So let's say that mom was the light-colored chromosomes and dad was the dark-colored. This cell is mom, dad, mom. The one that comes from the bottom chromosomes is going to be dad, mom, dad. You could also have mom, dad, 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 mom, mom, etc. in any combination of 23 sets of chromosomes. So you can see now that you are getting not only shuffled combinations of chromosomes, but you're also getting shuffled combinations of entire chromosomes. So the resulting cells at the end of meiosis is different because of crossing over and different orientations at the metaphase plate. This crossing over combined with random assortment is the greatest source of variation. So crossing over shuffles parts of chromosomes. Random assortment shuffles whole chromosomes.